leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid. But there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine. And these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first 2, and this process continues all the way down the mRNA. As tRNAs enter and exit the ribosome in a sequence that is dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached, at which point the completed polypeptide will swim away, most likely entering one of the cell organelles